Hi everyone. This video will examine how the genetic code works and the features of the code. In our last video, we saw how a complementary mRNA sequence was produced from a DNA template, so consider watching that first if you've not already done so. The information stored within the DNA sequence is first used to produce an mRNA sequence, which is then used to produce an amino acid sequence, which ultimately determines the final protein structure. This concept is referred to as the central dogma of molecular biology. The genetic code is a triplet code in that each group of 3 DNA bases within the coding section of a gene codes for a single amino acid. Within transcription, a DNA sequence is used as a template to produce a complementary mRNA sequence. Each set of 3 bases on the mRNA, known as a codon, codes for a specific amino acid. A table such as this can be used to determine which amino acid is to be added according to each mRNA codon. For example, if the triplet within DNA is GCT, the complementary mRNA sequence will be CGA. From the table, we can see that C as the first base in the codon selects the second row, then G selects the fourth column of this row, and the final base A determines the third line within this box, so the amino acid coded for is arginine. The first amino acid to be coded for is always methionine, as this amino acid is added by the start codon, which marks the start of the mRNA sequence to be translated. There's also 3 stop codons, marking where translation of the sequence will end. There are 3 key features of the genetic code that you need to know. Firstly, it's universal. This means that the same codons code for the same amino acids in all species. Secondly, the code is non-overlapping, so as the sequence is read in codons of 3 bases, each base only features within one codon, then the next 3 bases are read. Finally, it's said to be degenerate. The different possible combinations that the 4 bases can be arranged into groups of 3 gives us 64 different codons. Yet there are only 20 amino acids to be coded. Even when allowing for the 3 stop codons, which don't code for amino acids, it means that some amino acids can be coded for by different codons. In our next video, we'll look at how the mRNA sequence is converted into an amino acid sequence within the process of translation.